Hi, my name is Brandon Shah, and I'm from McGill University. I would like to thank the conference organizers for selecting this work for presentation. The title of my presentation is Mutation Edgotype Drives Fitness Effect in Human. This work is joint work with Mohammed Ghadi. A key area of investigation in network biology is to use protein interactome networks and other types of biological networks to make sense of the functional impact of mutations. Different missense mutations are known to cause different patterns of interaction or edge perturbation in interactome networks, and we call these different edge perturbation patterns edgotypes. For example, here we have the wild-type protein interacting with three other proteins. We also have three different mutations on the same protein, but with different edgotypes. The first mutation specifically disrupts the first edge, leaving the other two edges intact. This mutation is called an agetic mutation. The second mutation does not disrupt any interactions at all. This mutation is called a quasi-wild-type mutation. The third mutation causes the entire protein to fall apart and as a result disrupts all interactions associated with the protein. This mutation is called a quasi-null mutation. Mark Waddell and colleagues first recognized the importance of mutation edgotypes as the key link connecting genotypes to phenotypes. Indeed, through mutation edgotyping experiments, one can measure the precise patterns of edge perturbation caused by different mutations. However, what remains unknown is how these different mutation edgotypes quantitatively impact organismal fitness. We have some rough qualitative ideas about the fitness effect of different mutation edgotypes. For example, we expect that quasi-wild-type mutations are probably neutral, quasi-null mutations are probably deleterious, and agetic mutations are probably somewhere in between. Here, however, we would like to put these rough ideas on a rigorous foundation by quantitatively estimating the distribution of fitness effect for each mutation edgotype. How do we go about measuring the distribution of fitness effect for different mutation edgotypes in the human interactome? Well, we have to start from a high-quality human interactome. Precisely introduce one mutation at a time. Determine the precise pattern of edge perturbation caused by this mutation. Carefully measure the fitness change of the cell and the organism as a result of this network perturbation. Then repeat this entire process many times for all possible mutations. As you can see, this is a highly challenging experiment, so we need to find some other way to estimate the distribution of fitness effect for different mutation edgotypes. The approach we took is based on structural systems biology. The idea is to take the high-quality human interactome network already validated by experiments, then build accurate 3D structural models for a subset of these protein-protein interactions using homology modeling. In other words, we're not predicting new interactions. Rather, we're annotating known interactions with 3D structural details. There's a long history of successful applications of structural systems biology. For example, Mark Gerstein and colleagues used structural systems biology to study protein and network evolution. We used structural systems biology to study host-pathogen interaction and infectious disease. Haiyuan Yu and colleagues used structural systems biology to study genetic disease. So, this is an established technique known to provide reliable results. Here's how homology modeling works for protein-protein interactions. We start with a pair of proteins that are already known to interact from experiment. To build a 3D structural model for this query protein pair, we perform a sequence similarity search of the query protein pair against a library of protein complexes with experimentally determined 3D structures. If we're lucky, we will find a protein complex in the library whose sequences are similar to the sequences of the query protein pair. In that case, we can borrow the known 3D structure of that homologous protein complex in the library and use it as a template to build a homology model for the query protein pair. Once we build 3D structural models for protein-protein interactions, we can then use these models to predict the precise patterns of edge perturbation caused by different mutations, or mutation edgotypes. For example, here we have the wild-type protein again interacting with 3 other proteins via 3 different binding interfaces. We also have 3 different mutations on the same protein, and our 3D structure-based calculations predict that these mutations have different edgotypes. The first mutation is located at the binding interface mediating the first interaction, or edge, and is predicted to specifically disrupt the first edge, leaving the other 2 edges intact. So this mutation is predicted to be an agetic mutation. The second mutation is located elsewhere on the surface of the protein and is not involved in any binding interface. And as a result, it does not disrupt any interaction at all. So this mutation is predicted to be a quasi-wild-type mutation. The third mutation is located in the core of the protein and is predicted to significantly disrupt the overall stability of the protein. And as a result, the entire protein falls apart and all interactions are disrupted. So this mutation is predicted to be a quasi-null mutation. These mutation edgotypes can be accurately predicted by 3D structure-based methods such as FoldX and BindProfX. Here's our computational pipeline for using the structural interactome to predict mutation edgotypes. We started with 2 high-quality, experimentally determined human reference interactomes. The Y2H interactome, which consists of interactions identified in each2 hybrid, or Y2H screens, that are additionally validated by orthogonal assays, as well as the INTACT interactome, which consists of interactions identified by at least 2 independent experiments as reported in the INTACT database. From each of these 2 reference interactomes, we then built high-quality homology models for a subset of the reference interactions to construct the structural interactome. Next, we map different kinds of mutations onto the structural interactomes. These include common missense mutations not associated with any disease from the dbSNP database, as well as Mendelian disease-causing missense mutations from the CleanVar database. We then use the structural interaction information to predict the edgotype for each mutation. We predict a mutation to be agetic if the mutation occurs on the interface, mediating a specific interaction with significant change in binding free energy. On the other hand, we predict that a mutation to be quasi-null if the mutation is buried inside the protein with significant change in folding free energy. All other mutations are predicted to be quasi-wild type. We then repeated this edgotype calculation for all mutations. Here are the results. In the Y2H-based structural interactome, we find that 1.5% of non-disease mutations are agetic, whereas 12.6% of disease mutations are agetic. On the other hand, 1.7% of non-disease mutations are quasi-null, whereas 13.2% of disease mutations are quasi-null. In the INTACT literature-based structural interactome, we find that 2% of non-disease mutations are agetic, whereas 8.5% of disease mutations are agetic. On the other hand, 3.3% of non-disease mutations are quasi-null, whereas 22.1% of disease mutations are quasi-null. In short, only a small fraction of common non-disease mutations are agetic or quasi-null, whereas a much larger fraction of Mendelian disease-causing mutations are agetic or quasi-null. These predicted fractions agree well with the observed fractions from mutation edgotyping experiments by Mark Vidal and colleagues. Now, we can safely assume that common non-disease mutations are neutral and Mendelian disease-causing mutations are deleterious. In fact, disease mutations are probably mildly deleterious on average because there are also embryonically lethal mutations which are strongly detrimental, but they rarely show up clinically as disease mutations. If we make these assumptions, then the pie charts shown here tell us the probability of neutral and deleterious mutations belonging to each of the 3 Edgotypes. In other words, probability of Edgotype given fitness. But what we really want is the distribution of fitness effect for different mutation Edgotypes. In other words, probability of fitness given Edgotype. So we need to invert the two sides of the conditional probability. How do we do that? Well, it turns out that we can use the Bayes rule to invert the two sides of the conditional probability. We already have the likelihood term, P of Edgotype given fitness, from the pie charts in the previous slide. We need the P of fitness term, which is the prior probability for a mutation to be neutral or deleterious, but we can get that from the literature. There's also the P of E term in the denominator, but it is just a normalizing constant, which we can easily get rid of. So we have all we need to calculate P of fitness given Edgotype. Here are more details on how we apply the Bayes rule, focusing on a specific mutation Edgotype, namely agetic mutations. We obtain from the literature the prior probabilities for new missense mutations to be effectively neutral, mildly deleterious, or strongly detrimental. Next, we added the pie charts from the previous slide, which give us the probabilities for effectively neutral and mildly deleterious mutations to be agetic. Now, we're missing a third pie chart, which is the probability for strongly detrimental mutations to be agetic, or P of E given S, but we can set lower and upper bounds on this probability. This probability, P of E given S, may be as low as 0, which corresponds to the scenario where strongly detrimental mutations are all quasi-null rather than agetic. And it may be as high as P of E given M, which corresponds to the scenario where strongly detrimental mutations and mildly deleterious mutations share similar fractions of agetic mutations. The actual value of P of E given S is somewhere in between these two bounds, but we believe that it should be closer to the lower bound than to the upper bound. Now we have everything. We use the Bayes rule to calculate the probability of agetic mutations to be neutral, mildly deleterious, or strongly detrimental. We then repeat the calculation for other mutation Edgotypes as well, namely quasi-null and quasi-wild type mutations. Using this approach, we can estimate the distribution of fitness effect for different mutation Edgotypes. The top panel here corresponds to setting P of E given S to the lower bound of 0, whereas the bottom panel here corresponds to setting P of E given S to the upper bound of P of E given M. Like I said before, the reality is somewhere in between the two panels, but we believe that it should be closer to the top panel than to the bottom panel. As you can see, there is broad agreement between calculations from different structural interactomes. We repeated the calculation using experimental mutation Edgotyping data by Mark Liddell and colleagues and found that they are broadly consistent as well. We see that quasi-wild type mutations are mostly neutral or mildly deleterious, that the majority of agetic mutations are mildly deleterious, and that a significant portion of quasi-null mutations are strongly detrimental. We can further combine the top and bottom panels, which corresponds to lower and upper bounds, into one figure here, showing the combined range for the distribution of fitness effects for different mutation Edgotypes. As you can see here, we estimate that more than 40% of quasi-wild type mutations are effectively neutral and the remaining are mostly mildly deleterious. That more than 75% of agetic mutations are only mildly deleterious, and that up to 75% of quasi-null mutations may be strongly detrimental. To summarize, I hope to have convinced you that it is possible to quantitatively estimate the distribution of fitness effect for different mutation Edgotypes. We used a structural systems biology approach to estimate that while very few of the quasi-wild type mutations are strongly detrimental, the majority of the agetic mutations are mildly deleterious, and a significant portion of the quasi-null mutations are strongly detrimental. What is really interesting is that a similarly large fraction of agentic and quasi-null mutations are non-neutral. These results suggest that purifying selection is as widespread in intermolecular interactions as in intramolecular interactions. I would like to thank Mohammed Ghadi for a great collaboration on this project. Thank you very much for your attention.